And we are live.
Perfect.
So we start. Hello and welcome to Biologically Speaking. Good evening, good afternoon, good night, and good morning, wherever you are, whichever part of the world you are in. Thanks for joining. Today we have a very special guest, Dr. Desikan, who is a director at the Quantitative Clinical Pharmacology Department of GlaxoSmithKline, UK. He is a computational biologist of having around 12 years of experience in diverse fields of research. He did his bachelor's in chemical engineering from the Visvesvaraya Technological University. It was the MS Ramaya Institute of Technology, and it fell under the Visvesvaraya Technological University back then. He went on to do his PhD in bacterial membrane, toxins, computational biophysics, and molecular dynamics with Professor K. Ganapati Ayappa at the Indian Institute of Science in Bangalore. He was a postdoctoral fellow in computational immunology and infectious diseases with Professor Narendra N. Dixit at the Chemical Engineering Department of the Indian Institute of Science in Bangalore. He was a scientist, and then he was a senior research scientist at Certara, UK, where he specialized in immuno-oncology and vaccine optimization. He then went on to become a principal scientist at Certara, UK, and currently he's a director of the Quantitative Clinical Pharmacology Department at GlaxoSmithKline. He specializes in infectious disease and vaccine. His diverse research skills include mechanistic modeling, model-informed drug discovery and development. He is also specializes in molecular modeling and different kinds of softwares. It's great to have you here with us here today, Dr. Rajat Desikan. And without further ado, I would ask you to go ahead. We're so looking forward to your talk.
Thank you so much. Can you all see my slides? Okay. Does it fill the entire screen?
Yes, I can see that you're starting screen share.
Thank you so much for such a kind introduction. It's an absolute delight to be here and to be speaking to all of you.
It's an absolute pleasure for us as well.
Does the slide fill the entire screen? Or
I can see that you have started screen share. I cannot see the slide yet. Is it?
So let me just try and share again.
Yeah, you just.
How about now?
No, I cannot yet see the slides.
So I'm just trying something else. How about now? No?
No, I can't. Yeah, please try again. I think it's.
How about now?
So I think we'll give it a few seconds because it's showing that you've started screen sharing.
So I see that your screen sharing is paused. I think it is not under my control. Can you just
yeah, just
the thing that needs to be done?
So Sanjana, is the screen sharing option on? Can multiple participants share?
Yes, it is.
It is, right?
Apologies for all the technical glitches. I wish I could talk to you all in person.
No, we are sorry for all the technical glitches, but as we have learned after doing so many live sessions, so they're part of it. So all of you who are here with us, please bear with us. We're sorting this out. Can you just try once more, or?
So what I'll try and do is
try turning the video off one. Yeah, this was
now it's there.
Is this better?
Yes, we can see your slides.
Okay. So can you see the slideshow now?
Yes. Yes.
Okay. Perfect. So thank you all for this opportunity. As you all know, I work in the pharma industry and I specialize in vaccine development for infectious diseases. I mainly worked on COVID-19 for the last couple of years, but now I'm working on chronic hepatitis B and so on. And I wanted to share a few of the insights that mathematical modeling could generate for optimal COVID-19 vaccine development and deployment. So this is sort of how I organized my talk into 5 sections. The first 2 are introductory sessions, where I want to briefly touch upon the role of mathematical modeling in biology. This is fortunately increasingly not important because many people are already identifying the role of mathematical modeling and the insights that it can bring into infectious diseases as well as drug development. But nonetheless, I would like to share one of my favorite examples about how mathematical modeling did a huge impact with respect to HIV. And then I want to give a 1-slide introduction of sort to how vaccines work. And this is more or less to ensure that all of us are on the same page and that the talk is accessible to a broader audience. And then I want to talk about 3 puzzles. Puzzle number 1. We are still vaccinating people with vaccines that have the original Wuhan antigen, which is the spike protein from the Wuhan strain that came out in 2020 and 2019. But we now know that there are a whole bunch of COVID variants, such as Omicron, which escape previous immune responses. So the question is, should we change the current SARS-CoV-2 vaccines to match variants? And what can modeling tell us regarding this? The second puzzle is, can we optimize vaccine doses and the interval between doses for maximum immune responses? And the third puzzle is, can we mechanistically link immune responses with population level protection? So this is more or less answering the question, what level of antibodies or cellular immunity elicited by vaccines would translate to protection? And can we quantify the link? Can we say that X level of antibodies would result in Y level of protection? So I don't know if we will have time to go till puzzle 3, but I hope to at least complete puzzle 1 and 2 during this presentation. So why is mathematical modeling important? So before that, we should more or less discuss what mathematical modeling is. And different people have different definitions of mathematical modeling. I like the definition by Robert May, who was a computational biologist at Oxford. He put modeling as a way of thinking clearly, where we explicitly mention the assumptions. We explicitly mention functional relationships between various variables, and we explore how a system behaves. Now, why do we need mathematical modeling? The reason is that when we look at complex systems, such as biological systems and so on, they exhibit something called as emergent properties. Emergent properties are a behavior that can only be seen as a whole and not by looking at the parts individually. Here is an example. We can look at a single vibrating string. We can look at all the properties, the amplitudes, the vibrations, and so on, and we can categorize it ad infinitum. But it's very hard to predict how a bunch of vibrating strings, here a violin concert, would actually feel like or behave like just by looking at the property of a single vibrating string. This is because a beautiful music concert is an emergent property. And if you look at immune systems are the quintessential classic examples of complex systems. There are so many feedbacks. There are paradoxical interactions. So the same entity gets upregulated and downregulated by multiple other interacting entities and so on. So it's a complex moving system with many, many parts, and therefore absolutely not intuitive. And it's not easy to decipher the behavior of the entire system based on looking at individual components of the immune response. At least it is not simple. And this, in my opinion, is a classic example where mathematical modeling can help us think clearly about the eventual immune interactions. Now, an example where mathematical modeling really made a difference. Here is the disease progression for HIV. As you know, HIV or human immunodeficiency virus is the pathogen that causes AIDS or acquired immune deficiency syndrome. So this pathogen infects a key immune cell called as a CD4 T lymphocyte. And you can see that after infection, the T cell counts drop until they almost reach zero, at which point the system's immune system is disabled, and they can die of multiple opportunistic diseases. They basically don't have any resistance against bacterial infections or influenza or any other infection that we normally can fight off without any issue. So how does the disease progression occur? So there is an acute phase that lasts a few weeks and a chronic phase that lasts a few years. And you can see that in the acute phase, there's a steep decline in T cells, followed by, and simultaneously, there's a steep increase in the viral load as well. Viral load is just a quantification of the severity of disease. It's the number of viruses per ml of blood. Now, when the virus kind of hits by the time it hits a peak, the immune system mounts a response. So the viremia gets controlled. And at the same time, there is a little bit of recovery of the T cell pool. Now, this phase called clinical latency lasts a long period of time where the T cells decline slowly over multiple years. And the viral load is more or less constant. Now, it is at this point that the physicians had a conundrum. In this clinical latency period, should you give drugs to the patient or not? Now, we know that the drugs for HIV are something called as antiretroviral therapy, which rapidly reduce viral load by interfering with the viral life cycle. For example, here you can see that after the treatment was given, the viral load came down from about 1,00,000 viruses per ml to about 1,000 viruses per ml, which is a couple of orders magnitude reduction. The problem is that art has a whole bunch of nasty side effects, and it can make living quite miserable. So the question is now, in this period where the virus seems to be quiet, should the patients go for years of therapy with this harsh medicine or not? Or should they just be left to live their normal lives and eventually, AIDS takes over? Here is where 3 mathematical modeling papers shed light on this issue. By looking at very simple sort of models, and these are the only equations that I plan to show in the entire presentation. I want to focus more on concepts rather than the how-to of mathematical modeling. The researchers came to a stunning conclusion. So the model is very simple. We have uninfected target cells. Viruses infect these target cells, and they cause infected cells. Infected cells release more viruses. A really simple model. And this model could fit in vivo data both with and without therapy. Okay, so what was the important bit of the model is that they could parameterize the production of viruses per infected cell as well as the clearance of viruses. And from this analysis, they concluded that the so-called latency phase where nothing much seems to be happening is a state of all-out war with about 10 billion viruses being produced every day. It looks like nothing is happening because the immune system is furiously battling the virus, and it's trying to clear out the viruses as soon as they're produced, which means that the immune system needs all the help it can get. So the clinical recommendation was that ART should be started immediately upon HIV diagnosis. And there were, we know that HIV is a mutating virus as is SARS-CoV-2 and so on, which means that it can escape therapy. So, however, modeling showed that you could combine 3 drugs, and this so-called triple combination, known as highly active antiretroviral therapy, is very hard to beat, and it can extend life by up to 15 years. And this is my favorite example of mathematical modeling influencing so many lives by predicting immune responses to diseases. And in this talk, I hope to convince you that that similar deeply impactful observations can also be derived and applied for vaccines. Now, the part two of my presentation. So this is really a fundamental sort of slide on how vaccines work and in how infectious diseases work. So when we have an infection with a pathogen, the blue area is where there is no damage to the body, so there are no symptoms, disease does not get shown, and so on. Above the dashed line is where the pathogen actually elicits symptoms of disease. So typically, when we have an infection for the first time, there is very little immune response that is tailored towards the pathogen. And hence, the infection almost always goes into the disease phase. At this point, the body mounts both cellular and antibody responses that control the viruses. So essentially, T cells kill infected cells, which are the source for the pathogen in the case of viruses. And antibodies coat the virus and neutralize it, preventing further infection and so on. So these two immune responses acting in tandem but not only reduce the infection, but they also stick around for a long period of time. And this is called as immune memory. So that when a second infection happens, there is already a basal level of antibody sticking around and a slightly higher level of cellular memory compared to when the first infection happened. And this memory is primed to respond to that particular pathogen. So we see a quick boost of cellular responses, which curtail the infection before we even get disease. And this is how for example, this is why we get chickenpox once in our lives, because once we get the virus, we have immune memory that protects us more or less for the rest of our lives. Now, the issue with that is that the first infection almost always crosses into the disease area. And this need not be safe for most of the infections. For example, diphtheria or measles and so on. So the aim of vaccination is to introduce an attenuated or an inactive form of the pathogen, typically a fragment of the pathogen or an inactivated whole virus or something of that sort, which mimics the pathogen but is unable to cause disease. So that we stay in the blue area, but the same immune responses are generated. And this gives us protection against further disease by generating similar immune memory as an infection without the side effects of infection. Now, this is a really simplified caricature of how vaccines work. In reality, the immune responses underlying vaccines are a lot more complicated. I'm not going to go into many of the details here, but I want to touch upon a few of the details that are important for SARS-CoV-2 vaccines. For example, we have heard about lipid nanoparticle mRNA vaccines, which is Pfizer and Moderna and so on, and adenovirus vectored vaccines, which is the AstraZeneca vaccine, or the Sputnik, which is the Gamaleya vaccine, and so on. There seem to be a whole host of vaccine modalities. Now, irrespective of the modality, all the vaccines induce antigens. The antigens stimulate the immune response. Initially, they stimulate the innate immune response, which is why we get fever and headache and all sorts of adverse events. And eventually, the adaptive immune response, which is the T cell and the B cells, that result in both cellular immunity as well as antibody immunity and immune memory. Now, the modalities differ in the way they produce antigen. For example, adenovirus vector vaccines are replication incompetent vectors that infect the deltoid muscle cell where the vaccine is administered. release the DNA within the cells, which then gets transcribed and translated into proteins. Whereas lipid and nanoparticle mRNA vaccines directly release mRNA, which gets translated to proteins, and so on. So there are differences between inactivated virus vaccines and so on. But the basic principle is this, that the vaccine produces antigen. Antigen is nothing but a non-self or foreign part of the pathogen, which elicits an immune response. And we get both cellular immune responses, which are T cells, and antibodies and memory responses. So this is a whirlwind introduction to how vaccines and vaccine-induced immune responses work. So let us go into puzzle number 1. So the first puzzle is should we change the current SARS-CoV-2 vaccines to match variants like Omicron? The current experiments seem to suggest mostly no. Here is a paper that recently came out in Cell. It came out in May, I think. And they had they directly measured contrasted updated the Omicron vaccines versus the original vaccines in monkeys. So they took 8 monkeys and they went through. They were given Moderna vaccine at weeks 0 and 4. And this is to mimic how the vaccination occurs in humans as well. And then they waited for a long period of time until week 41. At week 41, they were either again given the original Moderna vaccine, which encodes the Wuhan spike antigen from the ancestral CO2 strain, or they were given an updated mRNA Omicron vaccine that has the antigen from the recent Omicron strain. Now, this graph is a little complex, so let me break it down for you. The two families of lines that we should consider are the black and the green. The black are the anti-Wuhan titers, which is the antibodies that recognize the Wuhan spike. The green are the anti-Omicron titers, which are the antibodies that recognize Omicron spike. The solid line is after wild type vaccination. So it's Moderna, Moderna, Moderna. The dashed line is after Moderna and Moderna, followed by the Omicron vaccine. All right. So there are a few surprising things here. Let me break it down for you. The first thing is that after the first series of vaccinations, which is at about week 6, you can see that the Omicron titers are a few orders of magnitude lower than the Wuhan titers. This is expected because Omicron is an immune escape variant, and it's a shapeshifter of sorts in the sense that the spike protein of Omicron evades antibodies that are generated by the original spike protein due to mutations. The second, and this is probably one of the most interesting aspects, is that the Omicron boost given at week 41 elicited similar titers as the wild type boost. This is non-intuitive. We would expect that a matched Omicron boost would elicit higher Omicron titers. The third surprise is that the Omicron boost elicited similar Wuhan titers, even though it is a different vaccine. And this is borne out not just in antibody titers, but also in virus neutralization assays, which is a functional assay where you take the antibodies generated in vivo and do in vitro neutralization assays with live SARS-CoV-2 virus and so on. And you find similar patterns. Now, this is not just one study. Other studies with beta variant, if you may remember this as the South African variant, which is also an immune escape variant, also showed similar results. So our question was, why do we see these surprising results? And can we integrate multiple experimental data sets via simple models to understand the rules for boosting? This was work done in an academic collaboration with Professor Rustama Antia at Emory University. And we have a preprint out. So if you have suggestions or criticisms, we would love to hear it. we crafted a very simple vaccine model. I'll walk you through this model. So we can have either one of two vaccines. The first one being the Wuhan spike vaccine, and the second one being the Omicron spike. Now, the Wuhan and the Omicron spike share a few epitopes. These are called C, or the common epitopes, but they also have unique epitopes, a W in the case of Wuhan spike and O in the case of Omicron spike. And you can have antibodies and B cells that are specific to these three epitopes. Now, antigen can be, so antigen and antibody interact with each other to bind various epitopes in the model. For example, this is free Wuhan antigen. A common antibody can bind both the Wuhan antigen as well as the Omicron antigen, while unique antibodies only bind the respective antigens. Similarly, B cells are activated when they are able to bind their respective antigens. For example, this Wuhan specific B cell can either bind to a completely free Wuhan antigen at the blue area, or it can bind to this complex where blue is still free and it can interact with the B cell and so on. And so we can get, B cell and antibody responses to common epitopes, to Wuhan specific epitopes or Omicron specific epitopes. And the Wuhan titers are nothing but common antibodies plus the Wuhan specific antibodies. And the Omicron titers are the common antibodies plus the Omicron specific antibodies. Because remember that the common antibodies can bind to both, antigen and should be counted in both the immune responses. So it's quite a simple model. And if you are interested in more of the modeling details, please take a look at the preprint. The first thing we do after we create a model is to see if it makes sense. So we first tried to see if the model can recapitulate experiments that were already made. Here is the first experiment, and these are all independent experiments carried out either in animal models or in human beings. So this is an independent experiment in Cell 2022, where they either gave 2 Wuhan vaccines separated by 4 weeks or 2 Omicron vaccines. Now, so after the 2 Wuhan vaccines were given, you would obviously anticipate higher Wuhan titers compared to Omicron titers. However, after Omicron vaccine, you would expect the exact opposite. You would anticipate higher Omicron titers compared to Wuhan titers. And our simulations capture this basic observation. In patients, you can see that after repeated vaccination, the antibody titers saturate. This is probably best captured in this Israel data set, where 4 doses of the Pfizer antibody were given. So you can see that after the second dose, the antibody titers start to wane. They get boosted by the third dose. They start to wane again, and they get boosted. But the boosted peak never seems to saturate, right? So if you just connect the line of the sort, and our model captures that. The reason is that once you have a high level of antibodies in the system, you get these completely bound state antigen, which do not activate B cells. So this is called as epitope masking, and this is a fundamental feature of vaccine models. Lastly, we also remember that infection and vaccination result in kind of similar immune responses, although there are a few differences which I won't go into here. So we were able to capture infection plus vaccination in a few individuals. So pink is vaccinated people who had Omicron breakthrough infection, and lavender is unvaccinated people who had Omicron breakthrough infection. And we were able to capture qualitatively the similar antibody profiles as them. So this is sort of model validation and proof that the model works reasonably. Now let us see if we can try and derive the rules for boosting. So the first thing that we tried to do was to recapitulate the data that we saw in the Cell paper. So just to show you again, the dark blue and the the dark blue and the dashed green are after 3 vaccinations with the original vaccine, and the dotted cyan and the solid green are after the updated vaccine boost. And you can see that it's designated as WWOM here, as opposed to WWW. So you can see that we recapitulate exactly similar observations. These are the experimental data, and these are the simulations. So if I may take you a little bit more in detail over the antibody profiles, you can see that the Wuhan titers after the first 2 primary series are high. Omicron titers are low because the number of Omicron epitopes are lower, and these are not immunodominant. But after the boost, when we give the Omicron updated vaccine, there is a little bit of difference, but not much. So this gives us a clue that perhaps 3 doses are not enough to evaluate. And this is just a reminder to show you that when we have immune memory, further infection results in a huge boost of responses. What happens if we give multiple vaccinations with the Omicron with the Omicron updated vaccine? Let us just follow the solid green line here. You can see that after the 3rd dose, which is the first Omicron boost, there is not much difference with the wild type. But as you keep boosting more and more, you can see that the green peak goes high. So in comparison with the Wuhan vaccine, which is the original, which are all the current vaccines that we have, the Omicron vaccine gives us higher and higher antibody titers. And the reason for this is that it is exactly what is shown here. The immune system needs repeated exposures of an antigen to boost its response. So we suggest 2 things. Firstly, the experimental design of all these studies that evaluate the Omicron booster must have an additional dose. Just 1 booster is not enough. Secondly, even for vaccination policies, where we think about giving boosters once in 6 months or so on to protect us against COVID waves, we might need to think about giving 2 doses for the vulnerable and the elderly. And what is the validation for this? So the validation comes from human influenza vaccine trials with a novel vaccine strain. Now, as you all know, human influenza virus evolves every year, and there are a few strains that that do not exist with that do not infect human beings. The H5N1 strain is one of these. So what happens when you actually vaccinate people with the H5N1 strain? It's like an Omicron vaccination in people who are previously either infected or vaccinated with a different strain. You see that after 1 dose, the stem, which is conserved epitopes. So let's just stick to this sort of nomenclature rather than biology. The black is conserved epitopes and the red is unique epitopes. You can see that after 1 dose, black shows a higher response, but this is inverted after you give a boost. So this shows the importance of 2-dose strategies, both to test our model predictions and to vaccinate the vulnerable and elderly for SARS-CoV-2. And we suggest that it is important to update vaccines. Now, even if we give a single boost and those people get the breakthrough Omicron infection, remember that it could be similar to having 2 vaccinations, which would give them probably better protection down the line. So this is puzzle number 1. I don't know how you want to do question and answers. I could take questions continuously, or I could also do it at the end.
So we have asked whoever is listening to type the questions in the chat, and after you finish, we can address them.
Perfect.
Okay, yeah.
Perfect. So this is the end of this section. I'll switch gears now to the second puzzle, which is, can we optimize vaccine doses and prime boost intervals for maximum responses? And this was done in when I was part of the pharma industry. So these are not mainly academic collaborations. Now, a brief introduction. Clinical pharmacology is a field that optimizes drug dose amounts, number of doses, and dosing intervals. For example, take antibiotics. There is a certain level above which the antibiotics are efficacious. And by level, I mean the drug concentration in either blood or any organ of your choice. And there is a concentration above which the drug becomes toxic. And this is called as the therapeutic window, the difference between the minimum effective dose and the maximum tolerable dose. Now, when we take one dose of antibiotic, let us say, or a drug, the concentration of the drug shoots up, but then the drug gets cleared by our body, either through renal filtration or liver metabolism or through many other routes. So the concentration of the drug falls. Now, when the concentration of the drug is out of the therapeutic window, we know that we have to dose again and again and again. And we maintain this dosing until the effect of the drug is seen. For example, in the case of antibiotics, until the infection is cleared. After the infection is cleared and the symptoms are resolved, you can stop the treatment. So this is a basic field that is applied for universally across all drug development, except for vaccines. And this was quite surprising because vaccines are a major therapeutic area. And one would think that it is complete common sense to apply this sort of optimization for vaccines, but apparently not. Here is an example with SARS-CoV-2 vaccines. Now, you will see that there is a huge discrepancy in the doses. For example, if you look at mRNA vaccines, which are, for example, Pfizer or Moderna, Pfizer uses a dose that is less than 1/3 of the Moderna dose. But if you remember, both of them have similar efficacies against the Wuhan strain, which is about 95% each. And if you look at protein vaccines, you see an even higher discrepancy. This is a 5 times discrepancy between two different vaccine companies. And it's not clear as to how different doses were chosen. So if you look at Moderna and the Oxford AstraZeneca vaccine doses, they were chosen because, similar vaccine platforms, but with different pathogens. For example, in the case of Oxford AstraZeneca avian flu or Ebola vaccines performed similarly at the same dose. So the same dose which worked for other vaccines were chosen for this vaccine, which is a little insane if you think about it, right? Can you do the same thing for, let's say, antibiotics? No, you can't. Each antibiotic has its own dose range. So if we look at what Barney Graham, the former deputy director of the NIAID Vaccine Research Center, and who was critically involved with Moderna vaccine's early development says, he says that's just how it's done. The whole point was to go quickly. Now, another leading company is called Vaccitech, and the CEO of Vaccitech gave a statement that, in my opinion, is the favorite. It is a little colorful, but this is what he says. It feels like people just threw a lot of spaghetti at the wall and saw what stuck. And this is an important statement because what people look for is they take a guess of the concentration that a certain vaccine would produce, and if it falls within the therapeutic window, it gets through. So these vaccines are very, very safe. I want to emphasize that. So the vaccine doses are always below the toxic doses. However, we don't know if it is close to the MED or if it's optimal, or we just don't know. Now, there are real consequences to not selecting doses appropriately. Let us just compare Pfizer and Moderna doses for adults. Pfizer used 1/3 of the raw material. If we put it another way, Moderna lost about 3 times profits, or they could have produced 3 times the number of vaccines to vaccinate many, many more people in the world. A few more examples, but this time in Moderna's favor. So sometime in mid-2021, we know that all the major companies were interested in pediatric vaccines. So both Pfizer and Moderna started, pediatric clinical trials with younger kids and so on. Now, in a small pediatric clinical trial, Pfizer gave a pair of 3 microgram shots. This is 10 times lower the drug amount as adults. And they saw similar efficacy in kids compared to adults. And they also saw better safety. And because the doses were low, the many doses did not trigger severe fevers and so on. So they went ahead with this dose. However, in a later stage clinical trial, which had a much larger sample size, this trial failed simply because of insufficient protection. The kids who were vaccinated with this ultra low doses simply failed to produce enough neutralizing antibodies. Now, at the same time, Moderna conducted another clinical trial with a 25 microgram vaccine. This was also reduced from 100 micrograms, but it was not as low as Pfizer's dose for kids. Now, they saw that this shot elicited the same level of immune protection against COVID-19 as a full 100 microgram dose in young adults. So Moderna got a competitive advantage. They moved forward with global regulatory submissions, and Pfizer is now playing catch up by giving additional doses and so on of their low dose vaccine. So this begs the question, couldn't this have been predicted via mathematical models? And here it is at least a little more intuitive in the sense that the Moderna pediatric dose was similar to the adult dose used by Pfizer. So you could at least guess that Moderna could do well. But there are some really non-intuitive scenarios. Let me take you through data for the Oxford AstraZeneca vaccine, which is what was predominantly administered in India. So the standard dose for the Oxford AstraZeneca vaccine is 50 billion viral particles. But in the clinical trials, a lower dose arm consisting of 22 billion viral particles was also tested. Now, when the dose interval, which is the interval between the first and the second shots, was the same, the lower dose followed by standard dose trial did a lot better than the standard dose trial. And if you look at the efficacy in preventing symptomatic infection, it's 80% versus 63%. Now, you must all remember the flack that Oxford AstraZeneca got in comparison to Pfizer and Moderna vaccines, with people saying that it's just not as efficacious. If you look at it from a purely biological perspective, it's not obvious why. And one of the reasons could simply be dose optimization. They did not get the doses right. This could be one of the reasons why the vaccines exhibit lower efficacy. Now, here is another piece of data where the same standard dose was maintained, but the dosing interval between the first and second doses was increased from 6 to 12 weeks. And again, the efficacy jumped. So this means that this field absolutely needs clinical pharmacology kind of modeling for optimizing both doses, the number of doses, and the interval. Which, if I may remind you all, is what is done for every other drug modality out there. The therapeutic window is calculated, and we try and ensure that the drug concentrations remain within the therapeutic window. Why not for vaccines? So there are important reasons why vaccine models are not often used. One of them is that the vaccine modality heavily influences the antigen concentration and the antigen type as well. For example, you could use whole inactivated virus vaccines, which are, for example, polio vaccines or Covaxin in the case of SARS-CoV-2. You could use protein subunit vaccines, which are, for example, Novavax for SARS-CoV-2 and so on. And you could, of course, use adenovirus vaccines like Oxford AstraZeneca. You could use DNA vaccines like Zydus Cadila, or RNA vaccines like Pfizer and Moderna, and so on. Now, all of them have a different mechanism of action, and all of them have different antigens that they present. For example, the whole inactivated virus vaccine also presents other proteins that are available in the virus, whereas an RNA vaccine, for instance, would only produce the spike protein, which are the small knobs that you see on the surface of the virus. The second reason is that the vaccines stimulate almost every arm of the immune system. Your innate immunity, adaptive immunity, you name it, it's probably stimulated. And because of the complex cascade of events that occur between the antigen stimulation and the final production of immune responses, these are highly non-trivial to model. So you must be wondering, right? How do people select vaccine doses currently? The answer is that they use empirical methods. So they take mice, they test a range of doses and look at the vaccine response with time. So this could just be antibodies. They plot the vaccine response as a function of dose. And you typically get this sort of bell-shaped curve where lower vaccine doses are simply not immunogenic. Higher vaccine doses are also not as immunogenic because they either exhaust the immune system or they overstimulate and so on. So there is an optimal window which works well in mice. Now, this is translated into humans by very simple sort of scaling. So one scaling is you just translate you multiply the scaling factor as a ratio of body weights. So if a certain dose worked in mice, you could just say 70 kilograms divided by 25 grams, which is an average mice weight, would be the human dose. And you choose a couple of doses around this interval, test it in clinical trials, and pick one of them. That's not rational dose selection any longer, because we are not repeating this sort of experiment in humans. And we are not integrating the principles of clinical pharmacology. So when I was at Certara, AstraZeneca approached us, and so did Daiichi Sankyo, for building a vaccine simulator for COVID-19 vaccines. And I headed that exercise with the QSP group, with Pete van der Graaf and Andrik Kershek and so on. And we did build such a vaccine simulator for both adenovirus vaccines as well as lipid nanoparticle mRNA vaccines. I don't want to bore you with all the details, but essentially we built a pharmacokinetics model where a certain concentration of the vaccines elicit a certain concentration of antigen in various compartments. The antigen gets distributed to lymph tissues and blood and so on. And in the lymph, they activate the immune response. And the immune response was then tracked as a function of time. This included both cellular and antibody responses. And we could put in population attributes into the model. For example, young versus elderly versus pediatric populations, pregnant and lactating women, and so on. And we could now look at all of the immune responses rationally from a mechanistic perspective. So COVID ushered in a completely data-rich era. In opposition to other diseases, we have a wealth of information regarding all sorts of antibody and cellular biomarkers. I don't want to spend too much time on this slide, except to say that the model was calibrated with various different biomarkers. For example, antibody in blood, antigen pharmacokinetics in blood, which is the concentration of antigen as a function of time, IgM, memory B cells, effector CD4 T cells, memory CD4 T cells, you name it, it was probably included in the simulator, as well as affinity maturation. So we know that antibodies get better with time in the sense that they're binding to that particular, their ability to bind to that particular antigen gets better. And we captured this as well. Now we validated this. Almost all the data sets included here are for lipid nanoparticle mRNA vaccines, such as Pfizer and Moderna. And we validated this by independently predicting the responses for Oxford AstraZeneca vaccine, which is a completely different modality. And you can see that the predictions are overlaid on data. This is a single dose of vaccination and the antibody titers elicited within patients, as well as a third dose that we predicted long ago in 2021. So this gave us confidence in the model. And here is an illustration to show that because we were able to predict 3 doses, and remember, this data was not used to fit or calibrate the vaccine model. We could do additional prospective simulations, like seeing what a 4th dose would do, and so on. How did we apply this vaccine simulator? If you remember, the first vaccine approvals happened in December 2020 and Jan 2021. But there was an issue. So the clinical trials for both the Moderna, Pfizer, and AstraZeneca vaccines were conducted with either 3 or 4 week intervals between the first and second doses. However, a lot of countries, including UK and India and so on, advocated for longer intervals because they simply wanted to increase the first dose vaccine coverage among the population. This led to a hot debate between immunologists and the clinical trial researchers and the society experts, some of whom said that you should adhere to what the clinical trial parameters were, which is a 3 or 4 week interval. And others saying that it probably doesn't matter. It's more important to ensure that many more people get the first dose than people getting the second dose and so on. So this was a perfect opportunity for us to see how changing the dose interval would change the immune responses. Shown here are the antibody titers as a function of week and for different dose intervals. You can see that the dose intervals go from as little as 3 weeks to about 12 weeks. There's a huge difference between using a 2 or 3 week interval versus a 4 or 6 week interval. However, there is not much difference between using a 6 week interval versus a 12 week interval. And this is because the antibody responses saturate. In fact, this is a little bit of a bell curve with the optimum at around 7 to 8 weeks. So a 7 to 8 week dosing interval between the first and the second doses probably elicits the maximum antibody concentrations. We did this prediction in December 2020. We archived this result in Feb 2021, and we published it in July 2021. Interestingly, the pitch study in Oxford, it came to the same conclusions with the Pfizer vaccine, with the study chief investigator saying that 8 weeks intervals is a sweet spot. Now, here is a utility of mathematical modeling for vaccine responses. We not only said that predicted that increasing the dosing interval does not harm the vaccine. We also said that we could also predict the optimum dosing interval. And we can do more applications of this sort. For example, we know that the antibodies elicited by different people are of different magnitudes with huge inter-individual variability in vaccine responses. This is shown here in colored lines, which is from a Moderna vaccine trial. And you can see that this individual in green produces way lesser antibodies than this individual. About 3 times or 4 times lower antibody concentrations. So we can simulate virtual patients in with our simulator and figure out what parameter changes could actually explain the whole range of intervals. So each of the black lines you see here are model simulations with a slightly different parameter value. We call these as virtual patients. And we can then do a sensitivity analysis to see why some patients respond better or produce higher levels of antibodies and why some people do not. We can also answer really nuanced questions. For example, between the second and the third boost, there was a huge interval of around 6 months to a year, depending on which country you're in. And a natural question that arises is, if you give such a long interval between the second dose and the third dose, would the third dose act as if it's a de novo vaccine? We could do these simulations. For example, here shown here is a 3-year-long simulation where we let the antibody levels decay, and at the end of the second year, we give a boost. So this is the third dose. You can see that the antibody responses are comparable to after the second dose, which means that the immune memory has enough potency to respond to remember the primary vaccine series and to boost the antibody titers. We can also, if you remember the Pfizer dosing fiasco, we can also look at what level of dose is optimal and in various populations. So shown here on the left and the right are virtual clinical trials. So these are clinical trials that were completed on the computer between young populations on the left and old populations on the right, and at 3 different vaccine doses, so 10, 30, and 100 microgram. So we simulated this for a virtual patient cohort, so which means that there were many virtual patients in the simulation. And we looked at the percentage responders. So this is sort of a survival analysis where a responder who goes below a certain threshold of protection is classified as having lost antibody protection. This is a simplistic example, but it illustrates how we can use mathematical modeling. So you can see that people who were given the 10 microgram dose quickly lost immunity within about 1 to 2 years, whereas probably higher doses led to higher immunity. And this is, of course, these simulations were extrapolations of these clinical trials. And there is another important difference. You can see that between the young and the old, the 30 and the 100 microgram dose work kind of equivalently. But with the lower 10 microgram dose, you never reach 100% response. This means that you can do rational dose optimization. And probably say that perhaps for the elderly, you need a 100 microgram dose, and perhaps for the young, lower doses could work. And this sort of dose stratification is important in the when we have lower number of vaccine supplies, as was the case for COVID. So hopefully I have convinced you that we can use mathematical modeling to answer clinical pharmacology kind of questions. Which is really, what is the dose amount, what is the optimal dose interval, and how many doses to elicit optimal responses. Now, I want to continue on to one more academic collaboration where we try to answer, or rationalize this data that I showed you earlier. So the framework that I presented before could not explain why a lower dose would probably result in enhanced efficacy. So if you remember, this is the lower dose, which is the 22 billion viral particles, followed by a standard dose, which is the 50 billion viral particles, compared with a standard dose regime with the same dosing interval. And here the dosing interval is different, but the dose amounts remain the same. Now, this was a collaboration with IISc and with my postdoctoral advisor, Professor Narendra Dikshit. And it's published here, so you could take a look if you wish. And to explain this, we had to go to a more complicated mathematical model of the vaccine response, where we consider events that occur in the lymph node after antigen stimulation. Now, I don't want to get into all the details, but I want to give you a sort of a big picture view of what happens after antigen stimulation. Once antigen is present in the system, there is competition that is created between the B cells to see which B cell would optimally recognize antigen. And this is important because B cells are the ones that secrete antibodies. And each B cell has a slightly different B cell receptor that recognizes antigens differently. So it is important for eliciting high affinity antibodies that we select the right B cells. So you can think about the lymph node as a sort of training school for B cells, where the B cells that bind to antigen at the highest potencies are selected. Now, how does this process happen? You can there are cells that are called as antigen presenting cells. Shown here is one example, and antigen is displayed on them sort of like fruit on a tree, if I may use that analogy. Different B cells try binding to this antigen to try and pluck the fruit away from the tree. And depending on the affinity of their antibody receptors to the antigen, they either succeed or not. So once this sampling of antigen is over, all the B cells are compete for T cell help. And these are T cells that are specific to that particular antigen. And they preferentially help the B cells that internalize more antigen, which means that they had stronger antibody receptors that could bind to the antigen. Whereas the B cells that fail to internalize enough antigen, or to continue using the analogy, fail to pick enough fruit from the tree, are killed. This results in high affinity B cells that constitute both immune memory as well as antibody secreting cells. And of course, this whole process occurs for many, many rounds, which means that the affinity of the antibodies increase over time. And this process is called affinity maturation. Now, what happens with a lower dose versus a standard dose is that there is simply less fruit on the tree, which means that the selection is more intense. So therefore, the more potent B cells actually get selected. Now, of course, this is an optima, because if you reduce the antigen levels too much, there is not enough food at all for anybody, and all the B cells die. So there is actually an optimal dose that elicits the most potent antibody response. And a similar thing happens with the 8-week interval. Sorry, here on the y-axis is the B cell affinity, and on the x-axis is time. And the vaccines were given at weeks 0 and 4. And you can see that the lower dose caused a huge difference in B cell affinity compared to the standard dose. Now, the same sort of thing happens over an 8-week interval as well. Now, we know that antigen reduces with time. So as the antigen reduces, the selection becomes more stringent, and the B cells compete more fiercely amongst themselves. So increasing the interval before we give another antigen dose into the system actually enhances B cell competition in that period, which is why you can see that longer dose intervals or lower doses can result in higher efficacies. This reinforces the point that we must integrate mathematical models with experimental data to not only optimize the doses, but also the dosing intervals. How are we doing for time? I might have taken up a lot of time, so I don't mind stopping at this point of time and just taking questions rather than going into the next part.
So should we move into the questions? We have a few questions.
Absolutely.
Okay. So thank you so much for this talk. This is such an enlightening talk, and this is something which is going to interest everybody. I mean, there is not one person who would not be interested in vaccines after the pandemic, right?
Absolutely.
So I have a few interesting questions. I'll read them out to you. Tech Software is asking that how do we find out the viral latency period, which was shown in the HIV model that you showed earlier?
Okay. So the viral latency period differs from person to person, and it depends on on both the immune responses as well as the strain of the virus. As I mentioned, the immune system is furiously battling the virus at that point of time. So people with weaker immune systems progress to AIDS more quicker, which means they might have clinical latency period of only 3 to 5 years, whereas people with stronger immune systems or a different, more tolerogenic strain of virus may take longer to proceed to AIDS.
Okay, thank you. And Dushant is asking that the idea of a simple vaccine model, it sounds great to him. And what is the feasibility, or what are the pros and cons when we integrate the population data instead from different eco-epidemiological zones? What are the pros and cons that would be there?
Okay, different eco-epidemiological zones. So you're probably talking about this model, if I understand correctly. So that is an important question for multiple reasons. One thing is that people from lower economic countries, for example, some of the African countries and so on, have sorry.
I'm really sorry. There's a dog outside my window.
No problem. So people from lower middle income countries may have completely different immune systems compared to people in higher income countries. For example, people in Africa and India and so on suffer from malnutrition. They are also infected with multiple diseases right in their childhood. So this may be either a positive or negative because the baseline immunity is different. These differences have to be accounted for. The other thing is that the basic immunology differs as a function of race and ethnicity. For example, the Black races do much better with controlling some of the diseases such as HIV compared to diseases in the European countries and so on. We know that pregnant women have lowered immune responses and therefore they are vulnerable for SARS-CoV-2 and so on. So the constitution of the population and the epidemiology have to be integrated with vaccine models and virtual patient populations to optimize vaccine deployment across different geographies. Does that answer your question? Sorry. Can you hear me?
Can you hear me now?
Yeah, I can.
Sorry, I was muted. And Shanchari is asking that is there a correlation with the future strain with our natural immunity built after getting infected with the previous strain? And is there a need to get booster every year to be safe from the infection? And before that, she has said that she really wants to know that after getting a view about the puzzle one that you discussed, that there's a remarkable lower antigen load for Omicron because it's an immune escape strain. So this is a question.
All right. So there are a lot of things to unpack here. Let me go to the right slide. For example, here, right? So when I said that the Omicron strain has lower titers compared to that of Wuhan, it means that the virus has assumed a different shape. It's like inoculating with one vaccine and measuring titers against a slightly different pathogen. You would expect the antibodies to be lower against a different pathogen because it was not what we vaccinated against. So this is not surprising. What is the real question is, when should you update vaccines. What's the extent of immune escape that would justify, updating vaccines to a new variant? For example, I would answer that the alpha variant did not need updating of vaccines because even though it also had a little bit of immune escape, it was much more similar to the original strain compared to Omicron, which is really almost a different clade. So I hope that answers one part of your question. So the second part of your question, if I understood correctly, is there a difference between vaccination and infection and future protection? Now, I hate to use the term natural immunity because vaccines also induce natural immunity. It's just a safer presentation of antigen without the side effects associated with diseases. So in my opinion, both infection and vaccines elicit natural immunity, but probably that's a different and more politically heated debate that's going on. Scientifically speaking, the infection it probably results in better T cell responses because a lot more proteins, such as the nucleocapsid protein, are presented to the immune response. So the expectation is that while the antibody responses are similar, you could have broader T cell responses. Now, this is, of course, muddied because like all infections, SARS-CoV-2 causes a blunted interferon response. It may blunt, adaptive immune response generation and so on. And that those are all studies in progress. But from a naive point of view, and this is true for other acute infections as well, infection could result in broader T cell responses. The downside is that the vulnerable have a higher chance of dying. And that, in my opinion, is not acceptable, which is why vaccination is the only way forward to ensure equitable and safe generation of immunity across the globe. I hope that answers your question.
So Shawnchari has another question. She's asking that, can we predict with mathematical models how many years we have to need to conquer this virus with our natural immunity? It comes back to the same place of natural immunity, like common cold. So yeah.
So that is a slightly more. I do not know if we will conquer this virus, in double quotes. The reason is that we have significant animal reservoirs of SARS-CoV-2. So it's not like we can imagine a hypothetical scenario where you could put the entire world in absolutely strict lockdown for a month. We will not get rid of the virus because there are animal reservoirs and so on. So if you look at it from a practical point of view, it's the mutation of the virus is unpredictable. There are multiple sources that will ensure transmission back into the human population, even if we somehow achieve sterilizing cure. So I'm not sure if we will ever conquer this virus completely. What we can do is like annual influenza vaccination, and that length is debatable. People are wondering whether it should be a few months or a few years. And I think it really depends on how quickly the virus evolves, which is not a predictable process. So the best thing that we can hope for is to keep updating our immune responses with periodic vaccination and hope that the eventual evolution of the virus goes towards milder strains like the other 4 coronaviruses that just cause common cold in humans.
Yes, fingers crossed. We are hoping that. So I would like to ask a question which is a little away from the biology part of it, but you talked about hesitancy for a bit, vaccine hesitancy, and the presence of animal reservoirs. So all of these are concepts which are known to us who's working in the field of biological research. But there's a lot of people who are not working in science, and for them, many of these informations, they come out as pseudoscience sometimes, because pseudoscience gets circulated a lot. There's a lot of misinformation, and we even called 2020 a misinfodemic, right? So it was, so much was out there. So how important do you think communicating the science behind vaccines is? As a vaccine researcher, as a clinical pharmacologist, how do you see the importance of science communication in this?
I don't think I'm really qualified to completely answer this.
It's just we would like to know your view.
Yeah, so in my opinion, unfortunately, generating misinformation is way easier than generating information.
Yes.
And it takes exponentially longer periods of time to combat misinformation with all sorts of logical rejoinders than to produce correct information.
Yeah.
Compounded with that is the fact that we are in the midst of an evolving pandemic. And while a lot of the insights that I shared in this presentation are retrospective, it is hard to make prospective predictions without acknowledging that there is uncertainty built into the current situation. There is nothing like a 100% surety at this point of time. Until we don't acknowledge that, it is hard to not to favor one camp over the other. I think that vaccine hesitancy can be combated by more effective communication by the governments and the CDC and all the other official agencies. But at the end of the day, it's natural human instinct to distrust or to believe in conspiracies and so on. And I don't know if we have a cure for that, to be honest.
Yeah, and it's I guess a place of uncertainty which leads to this gap, and more of misinformation comes out of that gap.
Exactly. So uncertainty can be interpreted in multiple ways.
Yeah.
And as a scientist, you cannot put your camp one way or the other, right? Because then it becomes belief and faith rather than science.
Exactly, rather than science. Absolutely. So with that, we come to the end of our session. I mean, thank you so much. It has been a great talk. It has been a great discussion. Thank you for answering all the questions. Thank you for being here, being part of this platform. And thank you so much. Yes.
Thank you so much for inviting and for all your questions and patience.
Thank you, all of you who are here with us today. Please keep looking at our Twitter account for the next events that are going to come up. And thank you for joining.
Absolutely. Thank you.
Thank you, Rajat. Sanjana? Sanjana live.
Just a minute.
Live talk.